All right, so now we are going to talk about how the immune system is regulated and talk a little bit more about tolerance. So we have, of course, we have our T cells and B cells that are going to be generated in primary lymphoid organs. And so, of course, it's the thymus for the T cells, bone marrows for the B cells. And we're going to undergo a process called central tolerance, which is negative selection in the thymus or the bone marrow. Now, keep in mind, we've already talked about this, and in the thymus, negative selection is under the direction of AIR, a gene called AIR. So it's the autoimmune regulatory. And so this gene gets turned on in the thymus to make a snapshot of your body in the thymus so that your T cells can undergo negative selection for antigens throughout your body. And so the negative selection will, of course, delete self-reactive T cells, and in the bone marrow, it will delete self-reactive B cells. So you can have individuals with a deficiency of AIR, so AIR does not get expressed. And these individuals tend to have a little bit of an increase in infections, but they generally live for a natural life. Now, another important aspect to tolerance is peripheral tolerance. And peripheral tolerance is mostly going to be by your regulatory T cells. And this is very important because if you have a deficiency of regulatory T cells, such as FOXP3, which is the transcription factor that is the master regulator of regulatory T cell genes, then your lifespan is only about 1 year. And so it requires a bone marrow transplant. So regulatory T cells as peripheral tolerance is really key to our survival. Now, other components of your peripheral or your peripheral tolerance is going to be immune privileged sites. It's going to be absence of co-stimulation or T cell help and activation-induced cell death. So we're going to talk about each of these, and we're also going to talk about regulatory T cells. Now, keep in mind, if the immune system goes unchecked, you get overactivation and damage. If the immune system is unregulated, you can lead to autoimmunity, and you can lead to also allergies, including severe allergies. And unchecked is what we talked about last time, is going back to homeostasis after an immune response so that your body can recover. Okay, so tolerance is in essence a situation where your cells have the ability to respond to something, but they're being told not to. So it's a state of unresponsiveness to something that they can actually respond to. And tolerance is not 100%. So there is leakiness. So central tolerance is tolerizing to self-antigen, and it's going to occur in primary lymphoid tissue. And so those are only the bone marrow and the thymus. And so for the bone marrow, we have our B cells, and they undergo negative selection, and they either pass or they undergo apoptosis. And then we have our T cells in the thymus, and they are going to, in the cortex, undergo positive selection. And then they're going to move to the medulla to go negative selection. And then they're going to be released or undergo apoptosis. Now, in the peripheral tolerance, this refers to all other tissues. And so we have, for peripheral tolerance, some longevity of our lymphocytes, which helps to prevent them being activated by self-antigens. So for our B cell, it's about a 5 to 8 day half-life. And for our T cell, it's approximately 30 days. There's also going to be immune privilege. And so immune privilege just means your immune system is not allowed in. There's also going to be regulation on activation. And so in this case, you might have a T or B cell that interacts with an antigen, but it doesn't see help or doesn't get that co-stimulation, and therefore it will go to become anergic. And anergic cells typically will upregulate fast so that it can be eliminated by an NK cell or another T cell. Peripheral tolerance is also going to have activation-induced cell death and T regulatory cells. Okay, so let's talk a little bit about each of these. Privileged sites, what does that mean? A privileged site is a site where you don't want inflammation, you don't want your immune system to go there. And it makes sense not to go there. So some examples are your central nervous system, your eye, reproductive organs, and all of these, your lymphocytes do not enter unless there's a reason for them to enter. And so also in these sites, you're going to have your epithelial cells expressing Fas ligand. So in essence, they're acting like a T cell or an NK cell. So if another T cell comes in and has Fas, these Fas ligand expressing cells can make sure that that lymphocyte gets killed. So the T cells do not typically circulate in these sites, and immunoglobulins cannot diffuse into the sites. Another way to prevent activation is where you get no co-stimulation or no T cell help. So no co-stimulation, keep in mind that in order for signal 2 to be given to a naive B or T cell, there has to be some sort of inflammation. For the B cell, this is carried out by complement. So when you do have an infection, complement will get activated, and that will provide that signal 2 for your B cells. For the T cells, it's going to be your antigen presenting cell that has signal 2, and this is usually achieved through activation of those pattern recognition receptors. Toll-like receptors, for example, will upregulate the co-stimulatory signal which will be signal 2 for T cells. And so if there is no co-stimulation, signal 2, or no T cell help with respect to B cells, then they're going to undergo apoptosis. And what's important to keep in mind is there's two things going on here. So let's just say we have our T cell. Let's draw a T cell, and we'll call this flu T cell. And then let's say we have a T cell cold. Okay, so they're different specificities. We have our antigen presenting cell that is infected with flu. And so this APC has MHC, and it has CD80. And this flu will have its T cell receptor that will bind to flu, and we'll have its CD28 that'll bind to CD80, and so this guy will get activated. Now here's a cold one, and of course this antigen presenting cell is expressing MHC with flu, which is this red, flu. And this cold one does not engage the flu peptide, but this also has CD80, and this also has CD28, but because it doesn't see signal 1, let me move this so I can draw it a little bit better. Let's go here. Okay, so this one sees signal 1 plus signal 2 plus, so this means activation. This one sees signal 2 plus, no signal 1. And so this one has nothing. So this would be no activation, no energy. Now if we draw another cell, let's see, let's do this. This is our T cell. And then we have our APC. And here we have our, look at a pink peptide. And this T cell receptor binds. So this is signal 1, yes. Signal 2, no. So this T cell becomes anergic. So your T cells can either see signal 1 with signal 2 and become activated, signal 1 without signal 2 and become anergic, signal 2 without signal 1 and nothing happens. And the reason nothing happens is because you're responding to flu doesn't mean you want to eliminate all your cold T cells or all your measles T cells or whatever it is. And so your T cells always will have that CD28 on the surface, can engage the beta 7 CD8086 on the APC side, but it doesn't mean they're going to get activated. Okay, and so the last one is activation-induced cell death, and this is pretty straightforward. So if a T cell sees antigen over and over, so it's a very common antigen, then it will undergo self fast ligand, or a fast ligand with another NK or T cell. So either one, but it will kill itself through apoptosis. Okay, so now let's go to regulatory T cells. Regulatory T cells can develop in the thymus, and they can become natural Tregs. That's what that N means. N, Treg is natural. And this develops in the cortex of the thymus, and no negative selection. Okay, so that's important. These natural Tregs do not undergo negative selection. They're released from the thymus as they become natural Tregs. And they go into the periphery. And so this Treg here is seeing antigen, the same as this CD4 T cell is seeing the same antigen, this pink antigen. The natural Treg will release IL-10, which will stimulate the T cell that is also seeing antigen, and this will suppress. The CD4 activation. Now, the Treg can inhibit many T cells. So one natural Treg inhibits many T cells. Now, there are some other Treg subsets that are being identified. We have now a Th3 and a TRI. but we're not going to focus on them because they're very new. But what we do want to talk about is peripheral Treg development, or inducible Tregs. And so inducible Tregs are interesting because inducible Tregs are a CD4 T cell that did undergo positive and negative selection. So your iTreg So what this means is, in theory, this T cell did see the snapshot of the body and did get tolerized to self-antigens. So the iTregs are very important for oral tolerance. Tolerance to antigens that are coming in through the gut that are not part of that air snapshot in the thymus. And so these can be food antigens, commensal bacteria proteins, and it's important to tolerize against them. And so in the gut, or the GALT, gut-associated lymphoid tissue, you have your epithelial cells that are going to express TGF-beta. And that's transforming growth factor beta. And they do this when they're in contact with commensal bacteria. Now, TGF-beta induces Treg formation. And they also are going to favor the production of immunoglobulin A by B cells. And so this is going to be more anti-inflammatory. Remember, in your gut, you don't want to cause a lot of inflammation. And all of this together is oral tolerance. So we have over here my little picture. So we have here a CD4 T cell that has its T cell receptor. It's interacting with MHC class II. It's seeing the antigen. The epithelial cells are coating the area with TGF-beta, and this is inducing the CD4 T cell to become an induced T regulatory cell. So just keep in mind that this induced T regulatory cell came from the thymus after positive and negative selection. And in the gut, you want to tolerize T helper cells because you don't want them to induce inflammation to food antigens, for example. So oral tolerance is being used to treat people who have hay fever, for example. And so what's basically happening here is that you're going to have sublingual, or under the tongue, immunotherapy to induce Tregs and cause a switch from Th2 to Th1 against grass allergens. So you're really trying to get rid of those IgE producing B cells and switching them to an IgG producing B cell. All right, and so the last thing on this topic is how much antigen is present, and that can obviously have an impact on what type of response you get and whether or not your immune system even responds in general. So the factor that we're discussing here is dose, exposure frequency, route, and whether or not there's co-stimulations. And so we have tolerance inducing and we have immune response inducing. And so you can see that tolerance favors a high dose of your antigen, whereas immune responses favor a medium dose. We don't have low dose here. For every single response, there is a minimum dose, and the minimum dose can be very, very different. For example, some bacteria are as low as 200 bacteria. Some are as high as 1,000 bacteria have to be present. So there's a difference between the types of antigens and when you start seeing an immune response. But in general, a very, very high dose of an antigen is going to push towards tolerance. It means you're seeing it over and over again. You're seeing grass, you're seeing a food antigen, something that's seen over and over again, and you don't want to induce an immune response to that. Exposure frequency, again, multiple exposures will favor tolerance, whereas few exposures will favor an immune response. The route is different. Oral is typically pushed towards tolerance because you're going into that TGF-beta environment, whereas if you have injections, intramuscular, subcutaneous, those are going to favor having an immune response. So you can get in bacteria that get into your skin through a cut. You can get mosquitoes that inject things into your blood, etc. Co-stimulation for tolerance is none. Co-stimulation for immune response is required. For a vaccine, this can be promoted by an adjuvant. So, for example, if you get infected with a bacteria, the co-stimulation initially is going to be those pathogen molecular patterns that are going to trigger those pattern recognition receptors. With respect to a vaccine, you're not giving a pathogen, so you have to create that co-stimulation. And you can do that with an adjuvant. So, for example, you can give a little piece of a bacterium that will trigger a toll-like receptor, for example. Okay, so that is the end of this topic. Thank you.